Aunque a veces me acuerdo. Hello, Evelyn. Es muy tarde por ahí. Aunque a veces me acuerdo de... Oh, it's time. All right, we have a lot to cover, so we're gonna jump straight into the nervous system. Does anybody have any questions before that? Not anybody? Okay, then let's start where we left off. We started looking at the electrochemical properties of a neuron. So one thing, just to put everything into perspective, the cells maintain a specific environment compared to their surroundings. They maintain a specific osmolarity. They maintain a specific ion balance. And we're gonna focus on neurons right now, but this applies to all cells in your body. All cells in your body will maintain a specific environment, a specific electrochemical environment that's different than their surrounding environment. So in that sense, remember the concept of homeostasis? The cell maintains homeostasis from the point of view of the cell. The cell maintains this electrochemical homeostasis. So one of the properties of cells is that they usually maintain a negative electrochemical potential compared to their surroundings. And this is a property we're gonna see in neurons. In neurons it is more noticeable because neurons, they undergo these changes in electrochemical potential that are interpreted as an action potential. We know neurons as an action potential, it's intuitive in neurons, but this process happens in other cells as well. Maintaining an electrochemical difference between the interior of the cell and the exterior environment. And the exterior of the cell. What's the exterior of the cell called? You're gonna hear it. It's called the interstitial space. The interstitial space. The space between the stitium, which is the space between cells, the interstitial space. So let's start with these electrochemical properties of the cell. We started viewing how sodium and potassium. We started viewing what happens to sodium and what happens to potassium whenever we open channels. So let's say And what we are basically seeing here is a membrane, and here are some ions, positively charged ions. Now, this membrane, the ions can't just make it across the membrane, especially if this is, let's say that this is a lipid bilayer. A lipid bilayer is hydrophobic. What does that mean? It means that charged substances such as water, they can't make it through. But substances without a charge, such as other fatty acids, such as estrogen, these molecules that are hydrophobic, they can make it through, but charged substance cannot make it through. So what's happening here? We have an electrochemical differential between these two. Let's say this is the inside of the cell, interior. I'm gonna draw a nuclei there, just to point out this is the inside of the cell. This is the outside. So there is electrochemical gradient here, right? You can say that the exterior of the cell is +4, or actually, let me do +4, 5, 6, 7. Exterior of the cell is +4, or you can say the interior of the cell is +4, or the interior of the cell is -7, based on this. Now, that's the difference in that's the electrochemical difference between the interior and the exterior. That is basically, however, if you open channels, let's say that there is a channel here that usually closed, there's a little door here. But if you open this door, now the positive charges can make it into the interior. They're going to go, again, with the gradient, they're going to enter the cell. So they're going to enter until they reach a balance, right? So 1, 2, 3, and let's say this one entered, and this one entered, and this one entered. These 3 entered. They're no longer here. Now there's somewhat of a balance. They're going to move until they reach a balance. This is what happens if there is only one ion present in this setup. If there are other ions, for example, in the cell, we have, in the interior of the cell, we have a lot of proteins. Usually, these proteins, they are negatively charged, or most proteins that are in the interior of the cell are going to have a negative charge. And there's just random proteins, proteins that help with the function. Lots of proteins within the cell. And overall, if you add the charges of the proteins, there's gonna be a negative charge. Now, proteins cannot readily move across the lipid bilayer, right? They're too big, they're charged. In order for them to move it, we have to pack them in vesicles to send them out. So they're not constantly moving in response to electrochemical changes, but the fact that there are proteins that are negatively charged, the cell itself already initiates with a negative charge due to the presence of these proteins. So this negative charge is going to play a role whenever we talk about these balances between ions. Let's say that once again we have this this setup of positively charged molecules. And now, if you have more negatively charged ions here, there's going to be more of them going into the cell, because they are driven not only by their concentration gradient, but now these negative charges are also pulling them. But that is, I wanted to highlight that as an example of what are the forces that are driving movements of ions into and out of the cell. We're going to look specifically at some of these ions that are moving into and out of the cell in response not only to their concentration gradient, but also in response to charges, that charge difference between the interior and the exterior of the cell. We saw usually the cell is at negative 70 millivolts, meaning that the exterior of the cell is going to be, that the interior of the cell is going to be more negative than the exterior. Negative 70 millivolts more negative than the exterior. This is in part due to the fact that we have these negatively charged proteins. We mentioned that they can't move. They're gonna be there. They're part of like function and structure of the cell. So the cell already starts with a negative charge because of this, that cannot really, proteins cannot move across the bilayer to equilibrate this charge. There's also the sodium-potassium exchange pump that's that you will see during, let's say that these ion channels were closed, the sodium ion channel, the potassium ion channel was closed. So the cell will be at its resting membrane potential of negative 70 millivolts. So once all of those, once we have these leak channels present, and these leak channels are going to allow movement of ions to occur. So this, I want to make a distinction between leak channels and voltage gated channels. So what is a channel? That I think that's something that we intuitively know. It's something that allows the passage of in this case, it's something that allows the passage of ions. A leak channel is constantly open, so ions are constantly moving. A voltage gated channel, its name is fairly descriptive. It means that it's a channel and it's gated, it's voltage gated. It means that whenever it reaches a specific voltage, that's when it changes, that's when it opens, or that's when it closes. So it's voltage gated channel. And we're going to see that changes in the electrochemical gradient, the electrochemical difference between the interior and the exterior are going to occur. That's how we respond to stimuli. Whenever something, you feel a touch, that leads to a change in voltage across a membrane, and that in turn activates voltage gated channels. It can activate sodium voltage gated channels. It can activate potassium voltage gated channels. What does a channel do when it's activated? It's open. Once you open a sodium channel, and there is this difference in concentration, is like a dam. Think of it as a dam, and on one side of the dam is a lot of potassium, or on one side of the dam is a lot of sodium. And when you open the doors of the dam, then the sodium just floats in. If you open the gates of the dam, the potassium just flows. It flows. And what's going to determine whether they open or close is going to be voltage, changes in voltage. Your cell, your neurons that are at a resting potential, whenever they're not being stimulated, they are at a negative 70 millivolts. What determines a negative 70 millivolts? We talk about the presence of leak channels. Ions are going to move until they reach a balance. Until they reach a balance, and that balance happens to be at negative 70 millivolts. The balance for all the movements of ions. Now, if I want you to keep in mind that role of these leak channels. One of the things that we saw here is the potassium leak channel. There is a potassium and there is a sodium leak channel. That means that potassium is constantly flowing. That means that sodium is constantly flowing. And we mentioned as an example that potassium, based on their concentration differences, it's going to flow, is going to reach a balance at negative 90 millivolts. So potassium is going to move, so that positive charge of potassium is going to move outside of the cell until the cell is at negative. We're going to lose potassium, we're going to lose positive charge until the cell reach negative 90 millivolts. So why is the resting membrane potential negative 70 whenever we have potassium channels open, leak channels, potassium leak channels open? Once again, we're making a clear distinction between leak channels and voltage-gated channels. Voltage-gated channels, we're gonna see them in action during an action potential. Leak channels, right now, we're watching them in action in basically their role in the resting membrane potential of the neuron. So why is the resting membrane potential of a cell negative 70 whenever we have potassium leak channels open, allowing potassium to move until it reaches a balance, and we have also sodium leak channels open, allowing sodium to move until it reaches a balance? Now, we talked about why whenever we open sodium channels, it goes from negative 70 to positive 66. Is because there's a lot of sodium outside of the cell, and that all of that sodium is going to want to rush into the cell. Why is there a lot of sodium outside of the cell, you ask? Because of this guy, sodium-potassium exchange pump. It's constantly sending 3 sodiums to the exterior of the cell. That is its job. Its job is to create these gradients. It uses a lot of energy, but without these gradients, we wouldn't be able to have this difference in concentrations. Without this difference in concentrations, whenever you pass a finger across the surface of your skin, you wouldn't have that response. Basically, whenever there's no gradient, it's like an empty cell phone battery. You pass your finger and nothing happens. But if it's charged, then if you cross your fingers, then the ions move. You have the ions can move from one place to another, and that movement is interpreted as an action. It's interpreted and can be translated. That movement of ions can flow from one neuron to another. If we didn't have these gradients, we wouldn't have a way to respond through this. We wouldn't have a current, basically, to send from one place to another. But anyway, sodium-potassium pump creates these gradients The leak channels allow these ions to move until they reach a balance. The balance is called the resting membrane potential. That's at rest. The membrane potential of the neuron is -70. Whenever if we look at the ions individually, let's say in a cell where there's only potassium, where the sodium channel is not where the sodium channel is not acting, where there's no sodium playing a role, you open the sodium channels and you open the potassium channel and the cell goes to -90 millivolts. That's whenever it reach. We mentioned there's a lot of potassium inside. The sodium potassium pump is pumping a lot of potassium into the interior. Then it's leaking out through the leak channels. And basically, we're going to lose this positive charge potassium until the cell is positively, it's at -70 millivolts. The question was like, why whenever we open both, and why do they reach balance at -70 millivolts? What not at 0 millivolts? Why not at positive 45 millivolts, closer to the equilibrium potential of sodium? Why at -70 millivolts, closer to the equilibrium potential of potassium? The answer is it's more intuitive. It's kind of intuitive. Basically, it's based on whenever you think about it, it's almost like a math equation. Why whenever you open these channels, why do you end up closer to what the equilibrium potential of potassium? It's because there's going to be either more potassium channels, or the potassium channel just sends more potassium to the outside. It's more efficient than the sodium channel. So that is why it's just a matter of quantity. If you have more potassium channels, then more potassium is going to make it to the outside, right? Compared to if you only have like one sodium channel, then sodium is going to try to go into the interior, but there's only one place to go to. So it's harder for the sodium in that sense to make it to the interior, whether it's going to be easier for potassium to make it to whenever those channels are open to rush into the exterior. So in a way, the equilibrium potential of the cell is closer to equilibrium potential of potassium, because potassium channels, leak channels, are more prevalent. So you can say that the equilibrium potential is mostly dependent on the potassium channel than the sodium channel. Now, it's not entirely, the equilibrium potential is not entirely at -90 millivolts. That's because the potassium channel is pulling. So basically, what's happening whenever you open sodium, positive charges go out, you lose positive charge, the interior of the cell loses positive charge. Whenever the sodium channels are open, the interior of the cell gains positive charge in the form of potassium. So positive going in, positive going out. The net balance of it is that there is more positive leaving than there is some coming back. Okay, so that it's understanding an equilibrium potential is challenging. So if you get lost at some point, please stop me, ask me any questions. We'll try to maybe explain it in a different manner, but don't be intimidated if we get lost. It's a challenging concept. So now maybe whenever we look at the action potential, maybe we can start understanding this better. So these changes in transmembrane potential, now we have a gradient. There's a difference in charge between the interior and exterior. What does that mean? That means that we have the potential to, there's a potential to create change based on not only the differences in charge, but the difference in concentrations of the ions between the interior and exterior of the cell. The difference in charge, sure, it helps because ions are not only going to be driven by their concentration gradient, but that charge also will either attract them or repel them. So here we have changes in transmembrane potential. There's going to be passive or active. This is what we talked about, the passive leak channels and the active gated channels. So here are gated channels. The leak channels are constantly open, and we mentioned the leak channels play a role in the resting in determining the resting membrane potential, because ions are going to move through these channels that are always open. They're going to move until they reach that equilibrium. That equilibrium is negative 70 millivolts. So it's like, think about ion musical chairs, maybe. Ion musical chairs is like, you open the leak channels are constantly open, and they're going to move, and they're going to reach a balance. And that balance is going to be based on what the concentration levels are going to be, what the charges are going to be, but that the overall gist of it is that it's negative 70 millivolts. The interior of the cell is negative 70 compared to the exterior of the cell. That's what it is. So there is more positive charges on the exterior than there is on the interior. Now, gated channels, on the other hand, unlike these passively leaking channels that are constantly leaking, they require a trigger. Usually you have here, this is the way that a neurotransmitter works. Wherever a neurotransmitter binds to its receptor, it can lead to a change. In this case, the change is the opening of acetylcholine, of this acetylcholine channel. This acetylcholine channel might be a sodium channel. So whenever this opens, sodium is going to rush in. So that these channels that you're observing here, they allow a specific ion to go through. In this case, in this example, they could be a sodium channel. So it opens and only sodium will rush into the cell. There are neurotransmitters that are associated with different channels, either a sodium channel, a potassium channel, a chlorine channel. So whenever an ion binds to the receptor, it will open. The channel that makes it through depends on what type of receptor that is. Is it a receptor that allows sodium channel? Is it a receptor that allows potassium? Is it a receptor that allows chlorine? In this case, this is, for this example, this is a sodium channel. So acetylcholine binds, channel opens, allows sodium. That's kind of how, and that leads to a change in the potential of the cell, right? It's a change in the charge of the interior of the cell. So it goes from, in this case, we have a voltage-gated channel. A voltage-gated channel is going to activate at a particular voltage. So, for example, positive charge rushing into the cell in this example, that's going to lead to a change in voltage. Whenever that voltage reaches a particular threshold, these channels will open. So these channels are made out of protein, and proteins are nothing than a positive and negative amino acids interacting with each other. Whenever the environment around them becomes more negative or more positive, the amino acids are going to react accordingly. They're going to have conformational changes because it's charges against charges. And in this case, once the ion reaches a negative 60 millivolts, so the cell, the interior of the cell has become more positive by 10 millivolts, it will open. The opening in this case, this is an example of sodium channel. Sodium rushes into the cell. Once again, these channels are very specific. They open and only one particular ion or substance makes it through. And then this particular channel, it closes at positive 30. So whenever it reaches positive, because sodium, we just allowed, by opening this channel, we have allowed a lot of positive charge to go into the cell. What's going to happen then? The interior of the cell is going to become more positive. Now the interior of the cell is at positive 30 millivolts. What's the resting potential of sodium? It's positive 66 millivolts. So if we open all of the sodium channels of the cell and we allow sodium to move against this to move until it reaches a balance, the interior of the cell will be positive 66 millivolts. What's the resting potential of potassium? The resting potential of potassium is negative 90 millivolts. That means that if we open all of the potassium channels in the cell, potassium will move out of the cell. So both sodium and potassium are positive charges. So what I want you to think about this is positive charges leaving. Positive charges will leave out of the cell until the cell is negative 90 millivolts. It has lost all of this positive. Now the cell becomes more negative. Whenever it's we were talking about sodium, we open all of the sodium channels. There's more sodium in the exterior of the cell, so sodium wants to come in. That's if you don't understand the logistics of it, just understand this, memorize this. Whenever you open, there's more sodium. Memorize this. This is the cell. This is the nuclei. This is potassium. That says okay. That means that there's more potassium in the interior of the cell. Okay? There's a lot of potassium in the interior of the cell. There's more sodium in the exterior of the cell. That's all you have to remember. To know what happens when you open a channel. So let's say, so now you know, now you memorize that there's more potassium in the interior of the cell, right? We got that. It's right here. I open a potassium channel. What's going to happen? Potassium is going to rush out because there's more of it inside, right? It's going to go to the exterior. What happens if I open sodium channels? Sodium is going to rush in because there's more sodium in the exterior of the cell. So I usually remember this as they saying, nah, the exterior of the cell, and the interior saying, okay. That's how you remember that more sodium in the exterior, more potassium in the interior. Those are the concentration gradients of these two ions.
I have a question.
Yes.
So on this example, the ion that's going in, which one will be again? Would it be potassium or sodium on the gated channels?
On this one, so you have to remember.
Not on your picture, but the PowerPoint.
Oh, the PowerPoint. Oh man, I forgot that this. I have the PowerPoint on, and my view is really small. My bad, you guys.
No, that's fine. Because I'm a bit confused, but I want to know what ion. I forgot which ion is on the PowerPoint.
So in the PowerPoint right now, it's like a nondescript ion. You see that it's just positive, right?
Yeah.
I said sodium
Okay.
because that's usually the ion, the positive ion that rushes into a cell.
Okay. So then why would the channel inactivated, why would it close at positive 30?
So the reason why it closes at positive 30 is it's kind of if it that's a particular nature of the channel. If it continue to be open, uh, let's say this we didn't have a closing mechanism. Let's say so this is the opening mechanism, the opening voltage,
Mm-hmm.
positive 60, and this is the closing voltage, positive 30. Let's say that we didn't have a closing voltage, and that the ion just remained open. So what's going to happen? This ion, sodium in this example, will rush into the cell until it reaches the equilibrium potential of sodium,
Mm-hmm.
which is positive 66, right? Um, however, the evolution or the system that was developed doesn't usually doesn't it closes the channel before it goes into that big swing. So there is a signal, there is a big change, as you can tell. It went from negative 70 to positive 30, and it will continue a little farther. However, it doesn't go all the way to positive 66, which is the resting potential of sodium. Uh, why your question was why would it close at positive 30? It's the mechanism where how it kind of um, the these gradients need to be reestablished, and you cannot reestablish a gradient if this channel remains open, right? The sodium potassium pump that's in charge of reestablishing these gradients, that's in charge of basically sending this gradient, this photo that I showed, the okay, and now more sodium into the outside, more potassium, more into in the inside. These are gradients. So that means there's a lot of sodium outside, there's a lot of potassium inside. Little sodium, little potassium here, little sodium in the interior. These gradients are being actively created by the sodium potassium pump. When in the example that you have in your in the PowerPoint, where we open the sodium channel, sodium rushes into the cell. All of this positive rushes into the cell until the cell is positive 30 millivolts. So that, but now that it's positive 30, that means, let me do another picture of that. What does positive 30 means? It means that now there's more sodium in the interior of the cell. The sodium potassium pump actively pumps the sodium out. So it can reestablish this gradient so that the action potential can happen again. Once again, you cannot reestablish the gradient if the channels remain open. If the channels are open, then you're pumping sodium outside of the cell and it's just coming back the same way it came. However, if you have a trigger, such as, okay, once we reach positive 30, we pumped a lot of sodium into the cell, close it up. Then this is closed, then we can reestablish, the sodium potassium pump can reestablish the gradient. Does that answer your question, Evelyn? Okay, good, good. Okay, so here we have mechanical regulated channels. Mechanical regulated channels basically are pretty intuitive. If you have a channel that whenever there's tension in the surface of the membrane, then the channel just naturally opens. It stretches and it opens. You have this happening in tendons. You have it in pressure sensitive areas. These are intuitive. This chapter is going to be heavy in voltage regulated channels. I'm glad we got in depth, we went in depth into that, because now we know how they work. We know that there is a trigger that activates it, and there's a trigger that activates it, and there is like eventually there's the same voltage trigger, a different voltage closes the channel. All right, so now those are the ones we saw. We're gonna talk about, we've been talking about potentials. What's a potential? A potential is like you have a lot of potential. Your lives are full of potential. What does that mean? The ability to do work. You have more potential whenever you wake up in the morning and you have a cup of coffee than you do at midnight trying to cram for a test. It's like you don't have that much potential. So that's whenever we talk about these potentials, I hope you understand them in those terms. It's like a difference. In the case of, for example, in the case of an electric dam, if let's say there is a lake here, and we put a wall, and we just build a wall in the middle of the lake. There is, and with this wall, and we have doors in this wall, in this giant wall, and then we open the doors. What's going to happen? Probably it's going to be a little more, a small amount of local movement, and that's because the water level is at equilibrium. There's really there's no potential to do work there, right? However, let's say that the lake is fed from only one area, and we close the doors, and then we allow the water to pool in one area, and then the second part of the lake, the water level starts getting lower and lower, and the water level on this place starts getting higher and higher. And just due to gravity and the concentration difference in water, now we have, and we open those doors, now water is gonna come rushing out. The potential, the difference of potential from case scenario 1 and scenario 2 are vastly different, right? Same goes for these potentials that we're talking about. These differences, or these differences in electrochemical charge that lead to the potential, the capacity to perform to undergo a particular change, in this case, an electrochemical change. We talked about an equilibrium potential so far. The equilibrium potential is the potential is negative 70 millivolts. It's the potential difference of the interior and the exterior of the cell whenever it's at rest. What determines that potential is at negative 70 millivolts? The presence of leak channels and sodium and potassium leak channels, and sodium and potassium rushing into and out of the cell respectively, based on their own equilibrium potentials, the places, the electrochemical difference at which they're in balance. The cell at negative 70 millivolts is in balance. That's as balanced as the cell is going to get. Now you understand that there is constantly, this is a dynamic balance. The sodium-potassium pump is constantly pumping sodium and potassium out to create these gradients of differences. And the leak channels are constantly open. Let's say if the sodium-potassium pump stopped working, but the leak channels are still there. What's going to happen? Sodium is going to rush in. Potassium is going to rush out to the leak channels. Slowly but surely, they will rush in. However, so these gradients that drive this difference in concentrations are going to eventually dissipate because the sodium-potassium pump is not pumping, not constantly rebuilding these gradients. If it stops working, potassium will leave, sodium will enter, and they will reach a balance. I can't exactly tell you where the balance is going to be. Closer to the potassium one, maybe negative 30 along those lines. But it eventually these these gradients will dissipate. So we talk about the equilibrium potential, and now we're going to talk about the equilibrium potential of the cell, the equilibrium potential of sodium, the equilibrium potential of potassium. Now we're going to talk about graded potentials. And what are graded potential? These are localized changes. This is a graded potential. Whenever a change in the electrochemical gradient in just one region of the cell. And there's the other potential that we're going to talk about are electric action potential. So we talked about the resting potential ad nauseum. These are graded potentials. These are localized potentials that are occurred, and if you get enough graded potential, you're going to get an action potential. We're going to see why that is. So here, the action potential is mostly going to happen at the axon helix. Why does it only happen at the axon helix? Once you understand the action potential, you'll understand why that is. This is the equilibrium potential. This is the resting membrane potential, negative 70. That's the sum of the equilibrium potentials of all ions in the cells. This is the graded potential. A graded potential is whenever let's say a neurotransmitter binds to this receptor. This is a acetylcholine receptor. This is a sodium voltage gated channel. So this not a sodium voltage changer. This is acetylcholine. It's a sodium channel. It's not voltage gated. I mean, it's just a acetylcholine channel. It allows sodium to go through. Sodium rushes into the cell. It changes the local environment. It went from negative 70 to negative 65, so it became 5 millivolts more positive, right? All of these positive entered the cell. So this localized change is a graded potential. So a graded potential will only change the local environment, right? Because this channel, can only affect this particular area right here. So in this example, it says all of the positive went into the cell, so it made this 10 millivolts more positive. Nearby 5 millivolts, and a short distance away, it didn't affect it. It's still at negative 70 millivolts. So these localized changes are, we were talking about neurons being communicating with a lot of neurons in their surrounding, and that the sum of all of those localized changes is going to determine whether a neuron fires or not. These are these small changes are changes in graded potential, localized changes of the environment, that if you sum enough of those, it will create this, the cell will depolarize. Why do we say depolarization? It's because the cell is polarized. The cell is at negative 70 millivolts. That's a polarized cell. If it was at 0 millivolts, that's not a polarized cell, that's a cell imbalance. But it's at negative 70 millivolts, that's a polarized cell. So effectively, whenever it goes closer, it goes from negative 70 closer to 0, is depolarizing. It's going back to a non-polarized state, basically. So whenever positive charge goes into the cell, it goes from negative 70. We mentioned it went to negative 65, negative 60, and it's depolarizing. These are the local changes that are happening. We talked about voltage-gated channels. Let's say if we had a voltage-gated channel that activates at negative 65, and that channel will open. Let's say that the channel opens at negative 60. So this hasn't reached that threshold to activate that channel, so it just goes back. It just goes back into its normal state. And that would be what's sending this back to its resting potential. Why doesn't it stay at this potential? This is where the leak channels and the sodium-potassium pump working together, they return the cell to that negative 70 resting potential. Okay? You have to understand the role of the leak channels. The leak channels basically, so what happened here? Like sodium entered the cell, right? But the sodium-potassium pump is constantly pumping sodiums out. So it sends all of the sodiums back out until we lost all of that positive out. Got it? So this is where if our sodium-potassium pumps stop working, then every neurotransmitter opening an ion and depolarizing the cell would just stay at this level. It wouldn't go back to the resting potential, so it'd be ready for another round of action potential, another round of stimuli. This is, the cell can also hyperpolarize. The cell can also, so what would make a cell turn more negative? This could be opening more potassium channels, allowing more potassium to leave the cell, more of that positive charge to leave the cell. We also have chlorine channels. Chlorine channels, chlorine is negatively charged. Chlorine, there's more of it in the exterior of the cell. We open those channels, more negative charge will make it into the cell, therefore making the interior of the cell more negative. We haven't gotten chlorine involved, but know that is something that happens. Okay, so here's the action potential. This is what happens. So there is a gradient potential in the localized environment. The gradient potential depolarizes the cell enough to open sodium voltage-gated sodium channels. Sodium channels open. What happens when sodium channels open? You have to say it in unison. What happens when sodium channels open?
Only sodium goes in? The channels, right?
Goes in. Yes. Sodium goes into the cell. That happens whenever sodium channels open, sodium goes into the cell. Whenever potassium channels open, potassium goes out of the cell. We talked about why do they go in that direction? It's because of the concentrations. Lots of potassium inside the cell, lots of sodium inside the cell. Open channels, sodium rushes in. Open channels of potassium goes out. Excellent. So we understanding that also allows you to understand what the changes in potential are going to be, because both of these are positive. So effectively, whenever we say sodium goes in, we're saying a lot of positive is going into the cell, right? So the cell is becoming positively charged. And whenever we say potassium channel goes in or goes out, we open potassium channels, it means potassium goes out. That means that we're losing positive, so that cell is going to become negatively charged, right? Okay. So, and we also mentioned that this, just like voltage opens them, voltage closes them. So at some point they're gonna close so they can return to this normal state. So that was the action potential. Let's look at the action potential in action. This is the resting state of the cell, negative 70 millivolts. You can consider the positive on the, what would this, if these were ions, what would the purple ion and what would the orange ion be?
Purple will be the sodium and the orange the potassium.
Excellent. You know that because there's more of it here, and you know that because there's more of it here. Excellent. These channels are closed at the moment because these are voltage gated channels. You see the gate here? Yes, they're closed. But if you reach a particular threshold of about negative 60 millivolts, you're going to open sodium channels. What happens when sodium channels open?
Sodium goes in.
All of this positive goes into the cell. All of that sodium goes into the cell. Eventually, eventually the cell becomes really positive. It reaches a positive 30. The channels close eventually at a particular, about positive 30. And at about positive 30, also what happens is that the potassium channels open. So all throughout, we're kind of, they don't open at the same time. They're like, after you, please, after you. Because if we have both of them open, then if we had both of them open at the same time, then sodium would go in and potassium would go out. So we're basically losing positive and gaining positive at the same time. That's not gonna lead to any change, right? It's gonna stay the same. But if we allow sodium to go in first, we're gaining all of that positive. And then we allow potassium to leave, then we're going back, we're losing all of that positive. But whenever we do, but in this case, potassium goes in, the cell becomes really positive because only potassium is moving in. But then at about positive 30, that's whenever sodium channels open and this positive leaves the cell until the cell actually reaches the cell reaches the resting potential of potassium at about negative 60 millivolts, negative 90 millivolts. And then potassium channels will close. Okay, so characteristic of action potentials generation is an all unknown principle. So either you reach the threshold for firing or not. Refractory period, it's a period upon which you can't fire an action potential until the previous action potential is complete. And why is this? In part is because of the cell reaching negative 90 millivolts. Now, whenever the cell is at negative 90 millivolts, before the cell was at negative 70 millivolts, right? In order to reach the threshold for firing, you only had to increase 10 millivolts. You only had to send 10 positive charges to the interior of the cell in order to reach the threshold for firing an action potential. Now that the cell has completed a cycle of an action potential, The cell it's more negative than its resting potential. So now, in order for the cell to reach the threshold for firing an action potential, is 30 millivolts away from it. So it takes a lot more. It takes a lot more change. In this case, what's causing this initial change? Usually, what's causing this initial change is usually signals, neurotransmitters, dopamine, touch of the skin. It localized graded potential. The short answer is graded potential. Small changes, activation of a channel that allows only of one particular sodium channel because it was activated by a dopamine, and it allows a localized change. And that change sometimes is strong enough to reach threshold, firing threshold. But now we are far away from firing threshold. We're negative 90 compared to the firing threshold of negative 60. So in order for this cell to reach the threshold for firing another action potential, there's got to be a lot of dopamine stimulating that cell in order for it to fire. So this is that refracting part, that refractory period, why it occurs. Also, yeah, this is in part. So now we have continuous propagation or saltatory propagation. The saltatory propagation is a characteristic of an action potential. It's basically jumps from, we talked about oligodendrocytes and shan cells myelinating them. They leave empty spaces in between them. These are called nodes of Rambier. And in these nodes of Rambier, they are rich in sodium and potassium voltage-gated channels. That means that you only need to activate these nodes of Rambier. Let me see where we are. Okay, there we are. See? Let me see if it's an example. There we are. There. Okay, this is the one where the myelination is showing, right? This is an axon. This is a myelin, and now in these nodes present here, here we have the sodium voltage-gated sodium channel and voltage-gated potassium channels. Here in this now you only whenever the wave travels from one place to another, you only need that localized change, that depolarization, that initial depolarization that reaches threshold, and those changes, you need it to happen at this level. Not throughout the entire membrane, but it happens here, and then here, and then here. So that's what's referring as saltatory conduction. That meaning that the changes in the membrane potential that occur whenever you open sodium channel, the changes in membrane potential that happens whenever you open potassium channel. Those are only occurring at the nodes, because those are the only places that contain the voltage-gated sodium channels, the voltage-gated potassium channels. And going back to the axon helix, why does the action potential initiate at the action helix? It's because the action helix has a high concentration of voltage-gated sodium channels and voltage-gated potassium channels in turn, because they do need to go back to their membrane, to their resting potential. But this is, once you reach the threshold for depolarization, the concentration of these channels allowed the action potential to initiate here and initiate in a strong manner. And then it jumps from one node to the next. It does its saltatory conduction and so forth. Okay, this is an example of there are axons that are myelinated and there are axons that are unmyelinated as well. The myelination of an axon and this saltatory conduction allows for faster propagation of a signal. But also we have unmyelinated action. Have you, do you, for example, this you intuitively feel the presence of unmyelinated. Unmyelinated actions are not as fast as myelinated actions, as myelinated axons. That tells you the role of myelin. But you feel it whenever whenever you hurt yourself. Have you ever hurt yourself and you feel an immediate pain, but then the pain that immediate pain stops, but then the dull pain initiates? That's because those two different types of pain are carried by two different types of axons. The immediate pain is carried fast by myelinated axons, telling you, ow, move. And the dull pain is there to remind you that you did something that you shouldn't do again. But the dull pain is carried by unmyelinated axons. They're slower. And this is a view of them. Their signal, a local current propagating and carrying, going down the axon. Myelinated axons, once again, the change only happening in between the nodes. But basically, you can think of an action potential happening at every single node. Depolarization reaching threshold at negative, going from negative 70 to negative 60, reaching threshold for an action potential, opening sodium channels that send opening sodium channels that depolarize the cell to positive 30, and then this opens potassium channels that sends the cell back to its negative 90 millivolts, and eventually sodium potassium bond returning to resting metal potential, and this current changes activating the channels next to it. The refractory period that we talked about is important because that gives the channel directional, that leads the action potential directionality, right? Because this signal, this action potential, this signaling membrane, these localized signals in membrane potential, it's happening at this level and it's happening at this level. But the action potential will trigger the change here and not go back the way that it came, because this is on that refractory period. This is on that period where the cell is more negative than the resting membrane potential, and it takes a lot more positive signal to send it to that depolarization threshold of positive 60. Axon classification type A, B, C, based on diameter, myelination, and propagation. Muscle tissue, higher resting potentials. Yeah. So cells in general, they have a resting potential. This is an example of those. Whenever we get to muscle, we're going to look in depth at the action potentials in muscles and to the propagations of them. Nerve impulse, presynaptic, postsynaptic, synapses. For the most part, you have chemical synapses, right? One neurotransmitter going to the next. But there are electrical synapses in which there is a continuous exchange of ions between one neuron to the next. They are rare, but the beauty of these synapses is they are immediate. They're fast, right? Because they're exchanging, they're continuous, so they're exchanging ions. And you can imagine the action potential just going from one neuron to the next immediately. Whereas in a regular neurochemical synapse, you have the action potential reaching the synaptic buttons, the end of the axons, and that fusing the neurotransmitters present there. Let's say that this is a dopaminergic neuron, it makes dopamine. So whenever the action potential reaches the end of the axon, it tells those vesicles to fuse, release that dopamine into the synapse. The postsynaptic neuron has receptors for dopamine, so that there will be a response there, changes in the graded potential in the postsynaptic neuron. And if those changes, the sum of those changes are high enough to reach, are strong enough to reach the threshold potential of negative 60 millivolts, then another fire action potential will happen in the postsynaptic neuron. But you see the complication of the process. Whereas these electrical synapses, though rare, they will be contiguous ion movement, an action potential that contiguously flows from one neuron to the next. These are rare and are found in some reflexes where you need immediate action. In some. Chemical synapses more common. There are, so what's going to determine whether a neuron will fire or not whenever whenever an action potential reaches the presynaptic neuron is the presence of whether the neurotransmitters are excitatory or whether they're inhibitory, right? Because you can send a signal that tells the neuron, the postsynaptic neuron, hey, fire an action potential, or you can send a signal that tells the inhibitory neuron, hey, don't fire an action potential. And what are those signals? The one that says fire an action potential means that it will turn the postsynaptic neuron more positive, closer to the threshold potential, to the threshold for firing action potential. If a cell becomes positive enough, and for the most part it's negative 60, it goes from negative 70 to negative 60. If a cell becomes positive enough, if a neuron becomes positive enough, it will fire an action potential. This is called the threshold potential. At this point is whenever it's like an all or none, it will fire. That happens if the neurotransmitter is excitatory, if it will make the interior of the postsynaptic neuron more positive. But there are neurotransmitters that are inhibitory, that will make the postsynaptic neuron more negative, farther away from the threshold potential negative 60. So you have neurotransmitters that will do that. GABA is one of them. Those are whenever we have during a weekend, whenever you go out for a drink, GABA neurotransmitters are inhibitory neurotransmitters are being released. So here's an example of cholinergic synapses, synapses that rely on acetylcholine. Synapses flow across the synaptic cleft. Synaptic delay is considered to neurotransmitter release, take appreciable amount of time. Let's look at the synapse. Acetylcholine is broken down. Acetylcholine choline is reabsorbed and recycled and reassembled. But let's look at the particular synapse. Now we're here. We're at synaptic. The action potential reach the axon endings. Now what's going to happen? Whenever the action potential reaches the synaptic endings, you're going to have, just like voltage, we have voltage gated sodium and we have voltage gated potassium channels. At the synaptic endings, we have voltage gated calcium channels. What's going to happen when the voltage of the action potential reaches the axon terminal? These voltage gated calcium channels are going to open. What does that mean? Calcium rushes into the cell. Okay, there's more calcium in the exterior of the cell than the interior of the cell. Calcium and sodium and chlorine. Calcium rushes into the cell, and calcium is what it does is it promotes the fusing of vesicles. Basically, promotes the release of the contents of these vesicles. What do these vesicles contain? Usually neurotransmitters. In this case, these vesicles contain acetylcholine. So the action potential reaches the axon ending, activates voltage-gated calcium channels, calcium rushes into the cell, calcium promotes, it's an imperative part of vesicle fusion. So these vesicles start fusing with the membrane. When they're fused, they release the neurotransmitter that they have inside of them. This allows calcium to fuse with the postsynaptic with these receptors. Eventually, what's going to come out of it? We are whenever the calcium fuses into these receptors, you're going to these are acetylcholinergic. These acetylcholine receptors are sodium channels. So whenever acetylcholine binds, they open and they allow sodium to rush into the cell. What is that going to do? It's going to make the postsynaptic neuron more positive. So this postsynaptic neuron becomes more positive. It might fire an action potential. Eventually, what is going to happen is this enzyme, acetylcholine esterase, you see this moon-shaped enzyme, is readily present in the postsynaptic synapse, and acetylcholine esterase is going to degrade acetylcholine. And here it is degrading. That way, it is ready for another action potential. These channels they basically have been cleaned and ready for another action potential. And what it does is eventually the cell is going to recycle the acetylcholine. Other neurotransmitter, norepinephrine, dopamine, serotonin, GABA, inhibitory neurotransmitter, dopamine excitatory, glutamate excitatory, serotonin can go both ways, norepinephrine for the most part excitatory. Neuromodulators, yeah, they can influence. So this is the acetylcholine neurotransmitter closer in action. So whenever we talk about these depolarization events that might influence a neuron to undergo an action potential, this is what's happening. Neurotransmitters, or in the case of your visual system, photons of light activating a particular receptor, and that receptor undergoes sends a signal. In this case, the signal of this particular channel is whenever it opens, it allows sodium into the interior of the cell. There are other methods how neurotransmitters function. They work through G proteins. They're called G protein coupled receptors. Their name is fairly self-descriptive. They're receptors coupled to a G protein. G protein coupled receptors. Here's the receptor. It's coupled to a G protein. Whenever it gets activated by a neurotransmitter, the G protein undergoes a conformational change that activates adenylate cyclase, that activates the second messenger system. What's the second messenger system? It's a system that allows the amplification of a signal. In this case, this neurotransmitter, whenever it activates one receptor. That means a one-to-one activation, right? But whenever this receptor in turn, it activates this adenylate cyclase, an enzyme that can turn many ATP, thousands of ATP molecules into thousands of CAMP molecules. Now this is a signal, thousands of CAMP molecules going and creating a change. Then that is this neurotransmitter creating its action. We have here nitric oxide is another neurotransmitter. Nitric oxide can readily cross the plasma membrane. It doesn't need a receptor. The reason why these neurotransmitters need receptors, ions need channels, is because of the lipid bilayer. They can't cross it. They have a charge. They can't cross it. That's why they need receptors. But there are other molecules that don't have a charge that will readily cross the bilayer. Nitric oxide is one of those. It just crosses it. And then it activates enzymes in the inside of the cell. It didn't need a receptor to do that. Just cross the plasma membrane, activated the enzyme. The enzyme produces uh, secondary messages, and it opens certain channels. So that's how it produces its change. Information processing, we talk about exhibitory and inhibitory and excitatory neurons. So these are two definitions of what happens in the postsynaptic neuron. Excitatory postsynaptic potential. That means that the cell, the postsynaptic neuron just became more positive. Inhibitory postsynaptic potential, it means that the postsynaptic neuron just became more negative. Whenever we mention that neurons are talking to thousands of other neurons, whenever you sum up all of the changes, the positive ions and negative ions that have the positive, the inhibitory and excitatory postsynaptic potentials, or whenever you sum all of that, that's going to determine whether the postsynaptic neuron reaches threshold. Negatives, it goes from negative 70 to negative 60 and to fire an action potential, okay? So there are terms you have to learn and understand, but I hope you can simplify them to know that inhibitory postsynaptic potential means the postsynaptic neuron just became more negative in response to a neurotransmitter, right? Excitatory postsynaptic potential, the postsynaptic neuron just became more positive in response to change a neurotransmitter. All right, so here's the first stimuli, second stimuli, threshold reach, propagation, information processing. Let's just dive real quick into it. This is an example of temporal and spatial summation. Temporal is kind of like you add, if a neurotransmitter fires and it fires again, then let's say if it fired the first time and it reached, the postsynaptic neuron reached negative 65 millivolts. If you wait enough time, everything returns to normal, the action potential fires again, then once again it's going to be reached negative 65 millivolts, the postsynaptic neuron. But if you fire right after the one firing right after the next, then it won't just reach negative 65, it probably reach negative 60, because you haven't given the neuron time for it to go back to resting membrane potential. This is what we were talking about way back here. Here, you haven't given the neuron time to return to resting potential. So if you fire a there is an excitatory postsynaptic potential that sends the postsynaptic neuron, it kind of depolarizes it a little bit. But then you fire one immediately, then that further positive charge is going to further depolarize the neuron. But if you fire that the firing happens, sorry, the excitatory postsynaptic potential happens here, and then here, then you're going to have just two of these curves, with none of which reach the threshold for firing an action potential at negative 60 millivolts. That is the temporal summation. Spatial summation is just like whenever you have two of them firing next to each other, so each one is creating like a local change in electric potential. So each one is making this postsynaptic neuron more positive, but if both of them are doing at the same time in the same space, then this positive is going to become way more positive than if they were doing it alone. So here, this is an example of excitatory action potential, and so this is like positive and negative, they can canceling themselves out. So you can think about this as a lot of positive going into the cell at the same time that a lot of negative, or in this case, right here, in this case, so what this is showing is like, hey, you can you can send a lot of positive into the cell, it will depolarize. You can send some negative into the cell, it will hyperpolarize. But if you send them both at the same time, an excitatory postsynaptic potential and an inhibitory positive postsynaptic potential, which is basically just making the interior of the cell more positive and more negative at the same time, then you don't have any change. GABA, so it's inhibitory. So these are examples of GABA releases on the axonal synapse inhibits opening of calcium channels as inactive node. Wow. So this is another level of this is another level of regulation that can happen. So we mentioned that an action potential reaches the axon endings, right? And usually that opens voltage-gated calcium channels. What happens whenever voltage-gated calcium channels open? Whenever open, calcium goes in. What happens when calcium goes in? Vesicles fuse. You have the firing of an action potential. However, sometimes GABA itself, there are GABA receptors at the axon ending, and there is this neuron over here releasing this neurotransmitter. And this is an inhibitory neurotransmitter. It will inactivate calcium channels. Basically, how does it inactivate calcium channels? It's an inhibitory neurotransmitter because it's like, remember this positive that was going, this positive action potential wave that was reaching? All of a sudden, this is you can consider, all of a sudden there is also a negative counterbalance occurring here through GABA and GABA receptors. These channels, these GABA receptors are channels that produce a negative environment. So basically, you're canceling out these positive charges coming through here. That means that you won't reach the threshold for these voltage-gated calcium channels to open. That means less neurotransmitter is released. So you're basically inhibiting this action potential. You're inhibiting the action potential at the level of the axon, or you can do the opposite. You activate the action potential at the level of the channel by basically firing an action potential at this level. But everything that we talked about, these localized graded potentials that causes localized changes that either depolarize or hyperpolarize a membrane or a neuron, that we kind of intuitively learn is happening at the cell body, it's happening at the axon helix, it's happening at the nodes of Rambier through an action potential. Now we're learning that can also happen at the level of the axonal endings. And once again, this could be excitatory or inhibitory. So it will make the action potential either more positive, it will make the signal more positive, the membrane. It will make the interior of the cell more positive, or it will make it more negative. Meaning that if it's more positive, then more voltage-gated calcium channels will open. If it's more negative, then less voltage-gated channels will open, and therefore, what does calcium going into the cell mean? That's another rule of thumb at this level, at the level of the axon, means that more vesicles will fuse and more neurotransmitters will release. That's presynaptic inhibition and facilitation. There is postsynaptic facilitation or inhibition, where it enhances presynaptic. Never mind. Okay, let me just, I think that's it. I think that's enough. That's enough. I still have to cover chapter 13. I might just make a video of it and post it, okay? Otherwise, we'll cover it. We're gonna actually, let's cover it Saturday, okay? Because I don't wanna make a video. I'm done doing homework. It's your turn. No, I'm kidding. We'll do it on Saturday where we can have questions and whatnot. Okay, thanks everybody for your patience. I know this time. Thank you for sticking around. All right. Bye.
Have a good day.
You too.